Hello, and welcome back to the Philip J. Curry Dinosaur Museum Virtual Speaker Series, where we have paleontology guests call in for a presentation on science, fossils, and dinosaurs, and where the public will be able to ask questions live on YouTube. First off, we want to acknowledge that we are on the homeland of the many diverse First Nation and Métis people whose ancestors have walked this land since time immemorial. We are grateful to work, live, and learn on the traditional territory of Treaty 8. Today, Dr. Oksana Vernygora is joining us from Lexington, Kentucky to present her talk, Fossil Clupeomorphs of Alberta. Dr. Oksana Vernygora is an evolutionary biologist with research interests focused on evolution and systematics of clupeomorph fishes, herrings, sardines, and relatives. She received her master's and PhD from the University of Alberta, where she studied evolutionary relationships of clupeomorph fishes using integrated analysis of morphological and molecular data. Currently, Oksana is a postdoctoral scholar at the University of Kentucky, expanding her skills and knowledge in the field of microevolutionary research, working on the population genomics of fruit flies. Thank you for joining us today, Oksana. And with that, I'll let you take it away.
Thanks so much, Cam. And thanks everyone for joining us today for our talk on fossil clupeomorphs of Alberta. So, as has been mentioned in my introduction, I did study herrings and allies for my master's, PhD, and my undergrad research. So they're of particular interest to me, but to introduce them a little more closely to everyone, I will mention that clupeomorphs are probably known to most of you, but maybe not by their scientific name. They're commonly referred to as herrings, sardines, anchovies, and overall we're talking about, roughly speaking, 500 living species and about 150 fossil species. The evolutionary history starts from the earliest fossils known from earliest Cretaceous, and the earliest known fossil that can be confidently put into Clupeomorpha is from Borneo of Brazil, and so it's about 130 million years old. Currently, we have our living Clupeomorphs, which are found worldwide, and they play a very important role in ecosystems, both in freshwater and marine waters. Modern clupeomorphs are very abundant and widespread, and they serve as the forage fish for other larger fish in aquatic ecosystems, also aquatic mammals, birds, and also for us humans who rely on them as commercial fish species. If you look at their distribution here on the right, you will notice one interesting pattern is that clupeomorphs are largely distributed along coastal lines of the continents. So their distribution traces continental coastal line very closely. And that's because preferred habitat of clupeomorphs are warm, mostly for most species are warm waters, but also relatively shallow waters. So they do like water depth less than 200 meters or so. So they stay close to continental shelf. And most clupeomorphs are marine fishes or fishes that spend at least part of their life in salt waters because they can migrate between fresh waters, brackish waters, and salt waters for reproduction, feeding, and other reasons. Very small portion of clupeomorphs are freshwater, exclusively freshwater species. In fact, only about 30 species are found exclusively in freshwater environments, and most of those fish are found in lakes in Africa, also in Amazon basin in South America, and a few in Indo-Malaysian region. So you can see that freshwater fish, freshwater species of herrings, they're a warm water loving species. They do like warm waters. You don't find freshwater clupeomorphs up north in high latitudes. But let's look a little bit closer to at what we have in Alberta, because that's where I did my graduate studies, and I was wondering, so what we can find there that's close to a local system. And so what do we have in Alberta? We have really nice aquatic system there. So there are lots of rivers and lakes, so which is suitable for freshwater fish to live in. And there are, in fact, about 65 species of fish, native and that have been naturalized in Alberta. But unfortunately, there is no herring. And one of the reasons for that is that if you remember what I mentioned earlier when we looked at their current distribution, is that freshwater herrings tend to be warm water loving. They like warm temperatures. And unfortunately, that's not the case in Alberta. So we don't have herring there. And even if we would have something that can migrate between saltwaters and freshwaters, unfortunately, it's a very long way from Alberta to the nearest sea or saltwater body. So some people may think, well, okay, no herring in Alberta, but what about lake herring? Because there is such a thing in Alberta as lake herring that some people would refer to. And lake herring, scientifically known as Coregonus artedi, is actually not an actual herring. Coregonus artedi is a member of salmon family. It's a salmonid fish. And if you ever wonder if lake herring, if you have lake herring, which is salmonid, or if you have an actual herring, you can look at their dorsal fin, dorsal outline, and what you will find in Coregonus is this adipose fin, which is diagnostic of salmonids. So that will be a giveaway that you don't have a true herring. So this is in a way a red herring. So this is a salmonid. That's why common names can be a little misleading. So what we have right now is that we don't have clupeomorphs in Alberta, but that's now. And we know that it hasn't always been the case because environment in Alberta has changed quite a bit throughout geological timeline. And so if we look at Alberta in the Cretaceous, because history of this group of Clupeomorpha stretches from earliest Cretaceous, so let's look what we have in the Cretaceous. During this time, Alberta was influenced by sea incursion that happened throughout mid to late Cretaceous, and this sea incursion created Western Interior Seaway that ran across the continent from north to south and covered most of Alberta, Saskatchewan, Northwest Territories, making them this shallow creating shallow aquatic habitat, which is suitable and very favorable for coastal fishes such as clupeomorphs. So this sea that was relatively shallow, and as I mentioned, relatively shallow coastal seawaters, preferred habitat for clupeomorphs. So that's why it wouldn't be a surprise if we find fossil record of clupeomorphs during that time in Alberta. When we look at paleo-environmental reconstructions of Alberta during Cretaceous, we see this lush paleo environments, beautiful vegetation and diverse animal life of terrestrial animals, as well as semi-aquatic animals such as turtles, crocodiles, Champsosaurus, and so on. But because during that time, a lot of the landscape was covered with rivers, streams, it was very wet environment, and when there is water, there will always be fish, or at least in most cases. And so during that time, aquatic environment was also full of diverse life, just as the terrestrial landscape. And fossil record of aquatic life was represented by diverse assemblage, but unfortunately, the main challenge of describing and identifying fish species from those assemblages is that most of them are rarely preserved as articulated specimens, and they usually when organisms die in rivers and lakes, they become disarticulated pretty fast due to either water turbidity, decomposition, scavenging, or combination of these factors. So articulated specimens, so fish specimens, are very rare to find. And most of them are known from disarticulated isolated elements, which you usually find in microfossil sites, and are usually known such as scales, vertebrae, sometimes teeth, or other elements of animals. Identifying and describing them is really hard work that is time consuming and also requires a lot of trained personnel, so people who know what they're looking at. Unfortunately, in Alberta, there is a team of qualified experts who work with this, and I was fortunate enough to meet them and work with them. And this team includes doctors Don Brinkman, Alison Murray, Julian Dewey, among others. So, if we look at the diversity of fish that we have during Cretaceous in Alberta, it's quite amazing. It's quite astonishing. We have sharks, we have rays, even non-marine representatives of this group, amiids, gar members of gar group, and sturgeon groups. Also, teleost fishes or more advanced bony fishes. And within that group, Telios, we have acanthomorphs and their relatives. So some of these fish are quite easy to identify from isolated elements because they're quite peculiar. And probably a lot of you, if you've been out in the field and looked at isolated elements, at microfossils, probably you will readily recognize some of them. So let's say for Rhinobatidae, for guitar fish, their teeth are quite characteristic. They have a flat surface, they have double roots, very peculiar elements. So those are usually come to easy to identify and place into taxon. Amiids are also quite distinctive in terms of shape of their vertebrae, as well as gars. These fish also easily identified by their jaw elements, by their teeth, and sturgeons, of course, quite distinct in a way that they have dermal ossifications that are also termed as scutes, and those have very distinctive pattern that can be identified. And another thing for sturgeons, sometimes you will find the leading fin rays that are also sometimes referred to as spines, but they're not true spines. Sometimes you will find those elements, and they're also quite easy to identify. But when it comes to Telios, so those more advanced bony fishes, true bony fishes that have all of their skeletal elements ossified, those become very tricky because diversity of their vertebrae and other skeletal elements is so great that without proper articulated reference material, it's really difficult to say for sure which taxon, is it a distinct species or is it just a variation within one species, or which elements should go together. So in that case, if you're interested in this whole field, so there is a whole different approach to deal with this problem when you just group them by similarity of their morphology and classify them like that until you have a proper reference material to assign those isolated elements to a specific taxon. For clupeomorphs, it's also the case. Unfortunately, clupeomorphs that we know from Alberta, a lot of them are also known from isolated material. So here is just an example of some isolated elements that were putatively assigned to clupeomorphs based on similarity with other well-known articulated specimens such as Diplomistus, but that's from Eocene, right? And also extant material. So don't forget that we also have extant material, extant Clupeomorph for comparison, but because so much has passed since between Cretaceous and current point, it's not always very helpful in comparison. But is that always the case? Well, fortunately, we do have some exceptional cases in which we find articulated, beautiful articulated specimens of Clupeomorphs, and that's the group I'm focusing right now, that provide not only perfect source of reference material to which we can assign isolated elements that we find elsewhere that are way more abundant than articulated specimens, but they also allow us to put those specimens, those new taxa that we described, into evolutionary analyses and infer relationships of these fish, as well as look at the diversity of fish in Alberta and infer distribution pathways for this entire group. So the first fossil that I want to talk about is a beautiful specimen of Horseshoeichthys armiceratus. It was a new genus, a new species described from Horseshoe Canyon Formation, which is very close, that was discovered from the same layer as the Albertosaurus bone bed. So that's just right there. This picture gives you a very wide view of the general area, so you can see, but those of you who've been to Badlands will probably recognize this general landscape. Specimen was recovered from Maastrichtian, so that's where Albertosaurus bone bed locality is, and it's located in the Dry Island Buffalo Jump Provincial Park. The age of this of this layer was estimated to be about 68.4 million years old, and originated in siltstone. So that's where the fossil was recovered from. If you've been to this area, and you're interested in fish, you will also know that very close to that general area, there is another very famous fish locality, which is called Pisces Point. It's younger in age. So if this locality from which Horseshoeichthys was recovered is about 68 million years old, Pisces Point, which I think I have a picture right here, Pisces Point is about 66, so it's very, very late Maastrichtian, and there are very interesting fish fossils that come from there, including oldest pike and some osteoglossomorphs, so very interesting site for fish paleontology. All right, but let's go back to Horseshoeichthys armiceratus. So here is the geological here is the geological formation from where it's coming from, indicated by the red arrow. And you can see that it's coming from the Tolman member, roughly saying Tolman member. And I know that some of my former fellow graduate students know, some of them are graduates, used to do field work about that general area. And it's just interesting for them, if they find any of the fossils that isolated elements that look like they could belong to a Horseshoeichthys or related Clupeomorph taxon, that would be great if they could refer to this material, compare it, and see if that's something that they find during their field work. This specimen came together with a few isolated elements, including right side, so the side that's not shown on the photo, right side of the rib cage, showing ribs, high mandibular, so that's the part that connects jaw system to the cranium, so a movable part of the skull to the fixed, unmovable part of the skull, as well as some vertebrae. Also some vertebrae from the main specimen, from the articulated part of the specimen, were extracted, were taken out from the specimen in order to study morphology of those elements. Because remember that articulated specimens are great reference points to compare isolated elements to. But the problem with articulated specimens is that sometimes, or quite often, I should say, they hide those features that are referred to when describing isolated elements. So if you look at the vertebral column in fish in this specimen, it's nicely articulated, and in some cases, it has ribs articulating with vertebrae. But the problem with that is that we can't really see ornamentation, articulation facets on those individual vertebrae, their shape. For that, we need to extract them and look in detail. And that's what authors of the study did. They extracted anteriormost vertebrae for the study in detail of those elements, and then being able to compare those elements to isolated microfossils that they find elsewhere in Alberta. And so what they found was really interesting. After comparison, they realized that a lot of the elements, a lot of the isolated vertebrae that were previously found in Oldman Formation, Dinosaur Park Formation, so those formations that are older in age, so Dinosaur Park would be 75, 76 million years old. In those parts, elements looked very similar to elements that they extracted from horseshoe ichthys from 68 million years ago. And so those elements were assigned to horseshoe ichthys, which allowed to age taxon to see temporal range of this taxon and how far back in time existence of it stretches. So that was very exciting, not only in terms of temporal range distribution, but also as a reference point for assigning microfossils to a certain taxon. For my work, because it was focused on evolutionary relationships, for me it was exciting because I had taxon that was found far, far north. So the thing with fossils, Clupeomorph fossils that come from Alberta, and also there is at least one from Northwest Territories, is that they represent the northernmost occurrences for this group. And that was the case for horseshoe ichthys. So when horseshoe ichthys was described and assigned to a particular group, that was in fact the northernmost occurrence of this group, which was at that point family Sardines before composition of that family was rearranged. And so after including this wonderful fossil that was very complete in terms of cranial features and vertebral features, after including it in phylogenetic analysis, what we found is that it fits within a family that within a clade that also contains very familiar to many people who are interested in fossils, taxa such as Diplomystis dentatus. And Diplomystis dentatus is famous because it's very abundant, it's very common fish fossil that's preserved. Quite often it's preserved as well-articulated fish from Wyoming, which is Eocene in age, about 52 million years in age, deposits from Green River. And so what we see is that horseshoe ichthys is placed in that group. And that's not surprising. There are definitely similarities in morphology between horseshoe ichthys and Diplomystis, but also it tells us that this group, this general clade, extends in time even further to younger ages. And by comparing isolated elements that we find in Alberta and that we were able to assign to horseshoe ichthys, temporal range can also be extended into Eocene to the time when Diplomystis dentatus occurred. Another thing is that is to think about paleo-environment and when and places, environments in which these taxa occurred. So for the older horseshoe ichthys armiceratus, who was found from about 65 degrees north, that's the estimated paleo latitude for this taxon, and it occurred earlier in time than Diplomystis dentatus. For Diplomystis dentatus, who is found from lower latitudes, temperatures, if we compare temperatures at that time, they would be quite similar. And so for horseshoe ichthys, temperatures, water temperatures would be about 10-14 degrees Celsius, and the same for Diplomystis. Diplomystis dentatus at that time. And that would indicate that with cooling temperatures, with cooling climate, this group followed this general cooling trend. They migrated south. They were pushed south because they have the temperature preference to specific value, to specific range. So that's also good to find this confirmation of the temperature preference in the fossil record if we're able to see this trend. Another thing that was possible to do, having isolated elements from a clearly diagnosed taxon, horseshoe ichthys armiceratus, and knowing its relationship to Diplomystis dentatus, was to compare the growth rate. And so authors who described horseshoe ichthys armiceratus did a very interesting thing. They compared growth lines on vertebrae in horseshoe ichthys and Diplomystis and estimated how fast those fish grow. They grew, and if their growth rates differ. And what they found is that for horseshoe ichthys armiceratus, growth rates were actually lower than for Diplomystis, and horseshoe ichthys was estimated to reach maximum size and peak of its growth at about 4 to 5 years. And then growth rate slowed down and was estimated to slow down and get to the plateau stage. For Diplomystis dentatus, the pattern was different. And for Diplomystis dentatus, as you can see, that growth rates were did not slow down even after 6 years of age, even at 7. They were still going at a pretty steep incline, indicating that Diplomystis dentatus was a faster growing fish and would reach greater size. And that's true because some of the specimens of Diplomystis dentatus are quite large in size, reaching up to 0.5 meter in standard length. So that would be from the tip of the snout to the end of the hyperoral plate. So that's a very interesting approach, how we can use information from microfossils once we have a good reference for them, what information we can extract. The same thing, so the same idea would be to age fossil fish, because the same approach that was taken to estimate their growth rates can be used to age fossil specimens that are found and see how old they are, which is very interesting. For that, in fish biology, the approach that's commonly used, just like with tree rings and in histology of other groups of organisms, you take elements, you cut them across, and you count growth rings. In fish, elements that are most commonly used for this approach to estimate their age are usually vertebrae, sometimes fin rays or fin spines are used, sometimes some of the elements of pectoral girdle, such as clythra, are used, and so on. In some groups, such as sturgeons, you can use scutes and count growth lines on those as well. So this is horseshoe ichthys armiceratus, beautiful specimen. And the second specimen that I want to present today is slightly different in the mode of its preservation. And I apologize that because I realized that not everyone is used to looking at fossil fish specimens, and compared to nicely 3D preserved dinosaur bones, sometimes it can be very difficult to wrap your head around how to look at fossil fish specimens. But here is a more typical preservation for a clupeomorph fish. It's very laterally compressed specimen. So what you see here is almost 2D specimen. On the left, you see the fish outline facing to the right, and the counterpart here on your left is facing to the left. So this specimen is a little bit older in age compared to Horseshoeichthyes armiceratus, and it was found from a black marine mudstone of about Cinnamanian in age, mid-Cinnamanian in age, around 95 million years, from Dunvegan Formation in Alberta, not very far from Grand Prairie, in fact. It's a beautiful specimen, fully articulated fish that's preserved and missing only distal parts of fins, of dorsal fin, anal fin, and of the caudal fin as well. It is described from, as you can infer from the name, is described from Dunvegan Formation from Cinnamanian right there. And just like with Horseshoeichthyes, it's pretty close to another famous fish formation and fish excavation locality is fish scale sandstone. That's a layer also in Cinnamanian from where diverse fish fauna is described. Interesting thing about this fossil is the way it was found and the way which is also referred in its name. So Tetraichthyes dunveganensis, the name etymology of this name is from tekeros, which from the Greek means lucky or fortuitous. And why that's the case for this taxon is because from what you see in this fossil, it looks like it was cut perfectly with a cookie cutter, right? It's just very nice circular in shape. And the reason for that is because it was taken during the core sampling. The core sampling went and cut core just perfectly to around the fish fossil. And so when authors were describing it, they decided to reflect that in a genus name, saying that it was very lucky that core sample was taken exactly around the fish. And you can imagine if it was left or right in any direction, that would only partially circumscribe the fish. But that's very fortunate. That was a very fortunate core sampling. And of course, ichthyes is from the Greek fish. This taxon is different from Horseshoeichthyes not only in the mode of preservation, that it doesn't preserve any 3D features in most of these specimens, but it's also different in a general shape of fish. So while Horseshoeichthyes is a more herring type body shaped fish, Tetraichthyes presents a very deep-bodied fish that is less typical of clupeomorphs, not only mostly today, because during diversity or modern diversity of clupeomorphs doesn't have a lot of deep-bodied fish like this, and most of them are more herring-like, more elongate and slightly laterally compressed. But Tetraichthyes is very deep-bodied, strongly laterally compressed. It has very strong spines along the on the ventral scutes, so scutes or modified scales that run along the ventral side or along the belly of the fish. They have very, very pronounced spines directed posteriorly, and also it has a very steep dorsal outline. So right between the back of the head and before insertion of the dorsal fin, you can see the steeply rising line, which is again not very typical of currently living clupeomorphs and some of the fossil clupeomorphs. But in the fossil record, from the fossil record, what we know is that there was a group of clupeomorphs of the fossil order Ellimmichthyiformes that exhibited this morphology. And that family is called Paraclupeidae. Paraclupeidae is known for this morphology. And so when this taxon was included in evolutionary analysis of this group of fossil clupeomorphs, it showed up as nested quite deeply. It was recovered quite deeply in the sclade of paraclupeid fishes of these fossil members of the group. If you remember, horseshoe ichthys was recovered on the almost opposite end of this large group of fish. So it's a very different fish and taxon in terms of its evolutionary relationships and morphology. So which is exciting because that tells us that there is so much diversity in terms of phylogenetic diversity and ecological diversity. So the close relative to Tachyrictis dumbiganensis was recovered to be Gastroclupea bernici from South America and also late Cretaceous in age. Those fish, as you can see, and that's what I was referring to, is that this group of fish is very distinct in their morphology. They have very deep bodies. They have very pronounced, very well-developed ventral scutes that cover more than half of the ventral sides of their belly, which is very interesting because we don't see a lot of this in current diversity of clupeomorphs. Maybe one group, Pristigasterids or long fin herrings, that would be an exception, but that's a relatively small group of clupeomorphs that we have right now. So with that, what we have and why these exceptional clupeomorph fossils from Alberta, why they matter so much and why they're so important in our understanding of clupeomorph and in general diversity of fish. It's because, as I mentioned before, both of these fossils, horseshoe ichthys armiceratus and Tachyrictis dumbiganensis, were described from the northernmost distribution of this fish. Another Canadian clupeomorph fossil, which is described from Northwestern Territories, from Albion. I'm not gonna talk about it, unfortunately, today, because it's slightly not from Alberta, is also the northernmost occurrence of this group. And what it tells us is that, first of all, the group was widely distributed worldwide, but also because for Clupea, taxon from Northwest Territories is from Albion, so about 110 million years ago. It tells us that by mid-Cretaceous, this group managed to disperse from where the oldest fossil is found in Brazil. It was able to disperse way up north, all the way here. So that gives us an idea of the limits of the distribution of this group at any particular point of time, being that Albion or being that late Cretaceous, as we have with horseshoe ichthys and Tachyrictis. And then connectivity of ichthyofauna, because for horseshoe ichthys, if you remember, it grouped in the same clade, in the same evolutionary group with Diplomistus. And we also have Diplomistus taxa from Eocene described from Asia. So there must have been connectivity, ichthyofaunal connectivity, connection, exchange going between North America and Asia, which is not surprising. It's obviously not news, but it's always good to find confirmation from different groups of taxa. So now we have evidence from clupeomorphs as well. And so with that, I would like to and with a slightly different topic. So how my research and my time during PhD and master's proceeded. So, and why description of fossils and paying more attention of really exceptional fossils matter. So during my time in the program, I was fortunate enough to join field work with dinosaur crews from University of Alberta that did field work in Dinosaur Provincial Park, and also other places, which was very, very exciting and definitely educational for me. I did not find personally any amazing articulated fish, which is fine, but I did work with some students who managed to do some amazing discoveries. And one of them, I do want to highlight this because that shows an example of wonderful student field work and in general student work that can be done and should be done more often. So one of them was an undergraduate student working at the university, studying at the University of Alberta, and also doing field work in Dinosaur Provincial Park. I believe that was in 2017. And he came across this incredible fossil. It's not a Clupeomorph, but still quite impressive. It's a sturgeon. It's a member of Accipensidae family. And the fossil is preserved in 3D, so it's three-dimensional fossil, missing posterior half of it. So unfortunately, we don't see pelvic fins, we don't see caudal fin, but head is there and scutes are there. There's so much of fishes there. And the fossil was described. So the student, Hiroki Sato, he found the fossil. He worked on his 499 research project, which is a research project that undergraduate students conducted at U of A, and I was helping him with that, as well as Dr. Philip Curry and Dr. Alison Murray, who supervised his research on this. And the high point of this was that his work was published in Journal of Vertebrate Paleontology. And to me, this shows how, first of all, that there is so many exceptional fish fossils that we yet to find in Alberta, but also that students can do all of it. This is an example where he found it, he described it, he published it. That was honestly a success story for a student, I think, and I hope this can be an encouragement to other undergraduate students and anyone who's interested in paleontology. And another example, another wonderful undergraduate student project that I was involved in, helping to provide advice and, I guess, opinion and help with descriptions, was project, again, a 499 project by Clarine Nebleza at University of Alberta, and she worked on describing sturgeon material found from Evapidea formation, that field work from which these fossils were discovered. And so again, she did her project under supervision of Dr. Corbin Sullivan and Alison Murray from the University of Alberta. And her project focused on taking those isolated sturgeon elements that are quite common to find. So they happen quite often, but it's just someone needs to sit down, describe them, and do something about them, because they can tell us a lot about paleo-environment and diversity of fish that we find. And so that's what Clarine did. And here in this picture on the bottom, she's comparing elements with extant material. So the extant sturgeon material is on the left and fossil is on the right. And she described texture, she described patterns of ornamentation, which in sturgeons is also quite distinct for different species. It's almost like, as you can see here, this is Superclathrem at the top left right here. So again, this is fossil at the top, much bigger than the extant material on the bottom right here. And texture of sturgeon elements, dermal bones or scutes can be diagnostic. It's not quite as snowflakes that everyone is each snowflake is unique, but in this case, it can be general pattern can be assigned to a species, and that's what she did. So I hope she. I don't think this project went all the way to publication, but it definitely provided some interesting reference material and information for future discoveries. And I hope in general, this effort continues, because knowing what those isolated elements are, and if we can describe and assign them to specific taxa, is very important, because that's what we work with. Okay, and with that, I would like to thank everyone who made all these projects possible. So I do want to thank Laboratory for Vertebrate Paleontology at the University of Alberta, where I did my master's and PhD, and where I learned so much about fish paleontology. My supervisor, Alison Murray, also Phil Curry and Eva Koppelhuis for inviting me to do field work with them, Don Brinkman for providing a lot of information about microfossils, and everyone who provided material and for these studies and research. And thank you. And I guess with that, I will take any questions.
Wonderful. Thank you so much, Oksana. That was an excellent talk. Some pretty impressive specimens for sure. So Lacerata Wang would like to say great talk. The fossil fish looks amazing. How might Tycheroichthys swim with that seemingly awkward body shape? Also on a tangent, which Clupeomorphs tasted best? Fossils included if it helps.
Okay, thanks for your questions. I will answer the hydrodynamic one first. So how did they swim with that shape? Well, actually this shape, the very laterally compressed shape, is not something surprising for fish. They're laterally compressed fish that you find, a lot of them, if you think about coral reef systems, right? Like butterfly fish, they're quite strongly laterally compressed fish. So that can be an adaptation to, in case of coral reef fishes, that's an adaptation for maneuvering through corals and other densely, habitat densely vegetated with vegetation or any other abstraction. So that could be adaptation to that. Another possible explanation for why that body shape, why we see that, is sometimes it can help with hydrodynamic qualities. So when you have high currents and strong flowing waters, let's say in river systems, that can also help being more narrow, more slender. That reduces drag in this respect. So that would be my explanation for that. The one thing is that environment in which Tycheroichthys was found is a little strange in that respect, because it was found in this deltaic environment. So it was estimated to be marine taxon. So it was estimated to be from salt waters, but it generally lived in the vicinity where there was incursion of fresh waters from river systems. And that's where exactly what might be happening is that you have high turbidity, you have high velocity of waters incoming. So it's not still waters. And that's why it could be that this body shape could be a way to adapt to perform better in terms of swimming. And to your other question, which Clupeomorph tastes the best, that's very subjective choice. I would say, in my case, I definitely prefer anchovies over herring. That's my personal choice. But again, that is a question for a debate, for a good culinary debate, I would say. Thanks.
That's a great question. Great answers
Yeah.
too. And yeah, so if anyone else has more questions, just leave them in the chat box and I'll read them out to Dr. Vernygora. Another question from Mermaid Labs. What will you be working on next? She also says, good to hear you again. Thank you again, Oksana.
Oh, thank you. So honestly, I'm broadly interested in my introduction, you heard that I consider myself evolutionary biologist, and in general, I'm interested in many things. I would say that Clupeomorphs is something that's very close to my heart, and I do plan to continue my work on Clupeomorphs. I still have, for my PhD, I have a phylogeny to publish. So yes, that Clupeomorphs will never be out of the view, definitely, of my research view. And yeah, so I hope to expand my research beyond looking at fossils only or at extant material only, and to have a more comprehensive picture. So maybe having a few research projects focused more on local adaptations of species or populations, how they adapt, because this is such a fascinating group. They have within the same species, they can have marine forms, they can have freshwater forms, and they can have migrating forms that migrate between freshwaters and salt waters. So it's honestly my interest in trying to see what, let's say, genetic basis for that ecological plasticity are. What are the basis why other fish are so narrowly adapted and other fish such as Clupeomorphs are able to migrate between those different habitats? Can we find explanations in their genes?
That is an excellent question to try and research. I'd
Yes.
love to hear the answer to that. That's a tough one. We have a question coming in from online saying, what is a dream fossil you'd like to find?
Dream fossil that I would like to find. Excellent question. I think it would be, so the problem with, again, it will be a Clupeomorph. It has to be a Clupeomorph. So I can answer that right away. But the fossil that I would like to find, I would like to find a well-preserved fossil of Alosidae. So that's another group. That's the extant group. So that's like shad group. And if you're thinking more, that's Alosa family, alewife. In, let's say, if we're talking about American shad, if we're talking about North America, I would like to find articulated specimen of that group, because when I had to do time calibrated analysis and trying to find a good fossil to calibrate my phylogeny with, that was a problem. In the review, it was always facing the same critique, saying that, well, whatever fragments we have, we can't confidently assign them to this group, to this genus, because it's just so fragmentary and there is no diagnostic features. So I would honestly like to find fully articulated Alosid. I know it might not mean a lot to everyone, but it's just a personal thing. So I know when they, and that would be like the earliest occurrence. That would be something from maybe Eocene, maybe even earlier than that.
That would be fantastic.
Yeah.
Mermaid Labs just wanted to add that last time I heard you talk, I actually ate some herring during your talk.
Oh, that's wonderful. And that's yes, that's why Clupeomorphs are just the best. I feel like if you can work on the group of organisms that are wonderful as organisms, there is so much about them to study, and you can also have them for lunch, then it's great.
You're enjoying them in all aspects.
Yes.
Someone wants to know, what is your favorite dinosaur?
Oh, what is my okay. I worked in paleontology group, so I should be prepared for this question. Honestly, my favorite dinosaur is, I guess, Triceratops. And that's yeah, in general, I just like Ceratopsian group because I do like I just like those dinosaurs, but I guess this whole passion for Triceratops is coming from my childhood when me and my sister would play Jurassic World video games, and there would be Triceratops. They would be like cause the most destruction running through structures and everything. So I don't know, to me, it's just this charismatic taxon that I remember from my childhood. And that's my favorite one, I would say.
They're an awesome group.
Yeah.
Did anyone inspire you to become a paleontologist?
Oh, that's a good question. If I was inspired, I definitely was inspired to become biologist. And that's the problem. I think I see paleontology as integral part of biology, of evolutionary biology in general. So I'm just so if there was influence, I definitely, honestly, I can say that when I started my program, my graduate program in paleontology, I was impressed by the amount of work. So I guess I found my inspiration. It's not that my inspiration came before I started my program, because I was generally interested in fish, and I started paleontological program because it allowed me to broaden my knowledge about evolution of Clupeomorph. So I very selfish in that respect. I just wanted to know more about Clupeomorph, not so much paleontology. But when I started paleontology, I guess my respect for paleontology and my inspiration to learn more in that field came from all that group, incredible group of researchers that I worked with, because honestly, it's an outstanding group of paleontologists at the University of Alberta. And I can say that lab I worked in terms of fish paleontology, my supervisor, my members of my committee, both master's and PhD. I had Dr. Philip Curry on my master's committee. I had Dr. Eva Koppelhuis on my PhD committee. I worked a lot with marine reptile lab, with Dr. Caldwell's lab. So, and Dr. Sullivan, who came in later. So all of this, I would say, this community in general just inspired me. And I would say that I consider myself lucky to be able to have that opportunity to work in that community, because I think this is just leading and in this whole field. So yeah, so they all inspired me. I guess that's just example of everyone who worked around me.
That's a great answer. Someone wants to know, the sturgeon that was discovered by Hiroki, was the back end missing due to erosion, or was it bitten off?
Oh, I think it was erosion in that case, because they did go back to that site later on. There was nothing. So it was an erosion, I believe, that destroyed the back end of the fish. Even though maybe it could be both, you never know. Maybe someone bit off the tail and then erosion just made it look like it's just an erosion. So
exactly. So if anyone else has any last questions, just put them in the chat. Otherwise, we'll wrap it up. And yeah, I think that concludes our question period. Thank you again, Dr. Vernygora. For those of you watching, join us again Saturday, June 19th at 12 p.m. as Dr. Aaron LeBlanc will be calling in from London to present his talk titled How to Become a Dinosaur Dentist, Making New Discoveries by Studying Ancient Teeth. Thank you again, Oksana, and thank you to everybody for joining in. Take care.